The plasma membrane has three names: plasma membrane, cell membrane, and plasma lemma. Now, with these three names, there is one thing which comes into account, that what is the function of plasma membrane. As we are aware that it is a selectively permeable membrane, so movement of any substance inside the cell or outside the cell, it is all dependent on the plasma membrane. Plasma membrane marks the separation between the outside environmental condition and the inside environmental condition of a cell. Or you can think of the demarcation between the surrounding and the cells inside. If there is any difference or any change in the outside environment, that will be notified to the plasma membrane, and ultimately the plasma membrane will respond accordingly. For example, think about a plasma membrane being kept in a hypertonic or an hypotonic solution. How will it react? Yes. So think about a hypertonic solution. A normal plasma membrane or a cell containing a plasma membrane is kept into a solution which is hypertonic. It means the concentration, it is a very concentrated solution. So the plasma membrane and the inside content, what will happen to the inside content of the plasma membrane? All the water which is inside will try to ooze out, because outside it's a hypertonic solution, and now the exosmosis is something which is going to take place. Similarly, if an already and what will happen to the cell? The cell will become flaccid. Similarly, this flaccid cell, when it is kept into a solution which is hypotonic or isotonic, then the reverse is going to happen. Endosmosis will result in the swelling or bursting of the cell. Now, because of that, the outside environmental conditions, they are affecting the inside biological membrane or inside subcellular content of the membrane in an eukaryotic cell as well as in a prokaryotic cell. It is not that only bacteria, bacterial cell plasma membrane is going to respond to outside environmental condition. Similar thing is going to happen with any other eukaryotic organism as well. Now, this entire idea of biomembrane or biological membrane came into existence first of all by Overton in Charles Overton in 1895. He suggested that the cell membrane was composed of lipids. What are lipids? They are fat containing molecules, and any substance which is fat containing, they understood, this scientist, he understood that any substance which was having fat or which was having lipid content within it, it could very easily enter inside a plasma membrane. But anything which was soluble in water, it used to take a lot of time to enter inside a cell, or at times it did not enter at all. The reason for that, please let me correct, this is a lipid. So according to his idea, any substance which is lipid containing can very easily enter inside the cell. So it means that like attracts like, and maybe the cell membrane is made up of lipid containing substance, and that's why only lipids can enter inside, isn't it? So this was the idea given by Overton, the primary idea, Charles Overton in 1895. Now, with the similar idea, if you think of most of the drugs or substances which have presence of drugs, when I say medicines, most of the medicine, they have outer covering of lipophilic uh, substance. This lipophilic substance allows the movement of the drug inside the cell very quickly. Had it been made up of, generally most of the pharmaceutical companies keep this idea in mind, and that's why they make the medicines in such a way that outer covering is a lipophilic covering, so that the movement of the drug inside the body will take place very fast, or there will be rapid movement. There is no dependence on any other ligand or receptor for the same movement. So that is idea which is used by cell membrane idea, which is used by the pharmaceutical companies for making the medicines. Second advancement took place in 1925. So you can see the gap, 1895 to 1925, when two Dutch scientists, E. Gorter and E. Grendel, they proposed another theory according to which the cell membrane was composed of double layers, which is a bilayer of lipid molecules. So there will be two layers of lipid molecules, and they measured that the surface area and the lipid content of the red blood corpuscles, which made it or which made them come to a conclusion that the lipid could occur only in two molecule thick layers. So other than that, there was no other possibility. There may be two layers, and that is the thickness in which there is presence of lipid molecules in both the layers, and that is the thickness of plasma membrane.